now. All right, guys. So let's talk about the structure of a nucleotide. Okay. So what do you guys know so far in terms of the structure of a nucleotide? So if you have your notes out, make sure they're next to you guys. Structure of a nucleotide. So what do you guys understand so far? What do you guys think?
There, there are three parts of a nucleotide.
Good. All right, but what is a nucleotide, Sophia? What do you think? What's a nucleotide? Or Valerie, or Elizabeth, whoever. What's a nucleotide?
A molecule.
It's a molecule, all right? So write that down, ladies. Write that down. So it's a molecule, all right? So we know that this molecule is made of atoms, okay? So what do these molecules look like, all right? So if we go back, okay, to DNA, okay, DNA is gonna look like this. It's almost like a twisted ladder, all right? So you guys need to understand that. So how do we build, how is this ladder built, okay? So that's what we wanna make sure you guys understand today, okay? So when we're looking at it, the big thing is if we take right now, this is kind of like a model, okay, of DNA. And so it's hard to see it. So what we're gonna do is we're gonna make it into like the models that you guys are used to with the atoms and the atoms and the bonds and stuff. So basically, what DNA basically looks like is when we take it and unravel it, it looks like this, okay? So now, as we're looking at it here, okay, what the big thing to notice is that, obviously, let's go here. Okay, how many sides do you see?
Two.
Right? Yeah, there's two sides, right? So when you're looking at DNA, right? So DNA, and let me go ahead and make this a little bit thicker here because it's hard to see. Okay, so when we're looking at DNA, right? Right? DNA has two sides, all right? So it has two sides. So number one, we got to understand that about DNA, right? And so as you can see, remember, Elizabeth, how many parts does a nucleotide have?
Three.
Three, right? And so as you're looking at it, right, I want to see, I wish I could give you guys access to my to my uh to my pen. But anyways, so what you guys can do, actually you can it? Let's see, annotate. Can you guys try to annotate? Does it work on your annotations? Are you allowed to annotate? Let me see.
I don't
Disable,
think so.
hide names of annotators. No, I think you can annotate. So if you go to hit that three buttons and hit more, I think you can annotate, I think. Maybe. Anyways. Okay, so um anyways, as you're seeing here, right? So I'm gonna circle right here. I'm gonna circle. Oh, let me get rid of this. Okay, so I'm gonna circle here, this. So see that right there? That right there is one nucleotide. Okay? All right, and as you're seeing, here's another nucleotide. You see that? Right? So there are two molecules, and now how are those molecules connected? What do you guys notice? How are they connected? Well, first of all, let's talk about the parts. So what's this circle? What do you think this circle is?
The phosphate group.
Good. And how about this one?
5 carbon sugar.
Very good. And then this one right here?
Nitrogenous base.
Very good. Now, remember, there in DNA, there's going to be 4 different nitrogenous bases. So nitrogenous bases in DNA, there's going to be 4. Adenine, thymine, guanine, and cytosine, right? And so right now, we don't know which ones of these are because we can't see the atoms, right? But as you can see, this nucleotide, how is this nucleotide connected to this nucleotide? How are they connected? Just look at the diagram. Just look at it. Just look at it. So what part of this nucleotide is connected to this nucleotide?
The 5 carbon sugar is connected to the phosphate group.
Exactly. See that? See that? And that's how nucleotides connect, right? And so if you take a look at this nucleotide here, right? So here's the phosphate group. Actually, that's not good. Okay, that you can't see that really good. Phosphate group, sugar, nitrogenous base, right? And if you see how the sugar is connected to the phosphate group of the other one. And so that's how these nucleotides are connected. All right, so that's how you build a strand. And over here, that's how you build the other strand, just like that, right? And so DNA, basically, it's built from all these little nucleotides. How many nucleotides are from right here? Okay, from here, from this line all the way to here. Okay, how many nucleotides are on the pink strand?
6.
6. All right, so look, we got 1, 2, 3, 4, 5, 6. Very good. 6. Now, which 6 nucleotides, right? And so now that's the next thing. Think of a nucleotide almost like a letter. Okay? So what I'm teaching you is basically the letters in the language of DNA. So anyways, but let's go ahead and let's roll this so you guys can see what I'm talking about, right? So now you guys kind of can see the nucleotides, right? And as we're rolling through here, okay, notice these are molecules, right? And so the molecules, okay, watch, you're going to be able to see them, right? Here we go. Hold on. Let's see. When they give you a really good diagram, we'll move forward. Let me see here, here, here. Okay, so all right. So now, as you can see, right? See what the only parts that change are the nitrogenous bases, right? And so now if we take a look, what are these what are these what are these circles right here?
Carbon.
Yeah, they're carbons. They're carbons, and they're basically atoms, right? So every corner is an atom. Here's a double bond, here's an oxygen, right? Okay, this diagram is not really good because this is actually I don't think this is right anyways, but at the end of the day, this thing here is a nucleotide. And as you can see, these parts, right, the phosphate group and the sugar, they never change. They never change. What's the only thing that changes?
The nitrogenous base.
Exactly, the nitrogenous base. So when we're looking at DNA, right, what we're looking at it, you can see as we zoom out, right, you'll see, oh, there it is. Now I can see it's backwards. So yeah, that's an oxygen. Very good. So boom, boom, boom. Yeah, so it's all good. See? So there's the 5 carbons. That's why it's called a 5 carbon sugar, right? Okay. This hydroxyl, that's the oxygen that we talked about in the videos prior, right? I don't know if you remember those. Okay. And then here we go. So as we zoom out, right? We're going to zoom out. All you really need to understand here, okay, let's go move out a little bit. Okay, we'll cover this later. Okay, here we go. And here we go. This is a good one. So as we're moving out here, okay, what's the only thing that's changing? What's the only thing that changes? What do you guys know? That's right, the nitrogenous bases, right? So this nucleotide here, right, has a guanine on it. Okay, this nucleotide over here has a thymine on it. Okay, and so when you look at DNA, all it is just a bunch of nucleotides, and the only thing that changes is the nitrogenous base. Got it? What do you notice is the different, what do you notice with the thymine? This is the thymine nitrogenous base, and this is the cytosine nitrogenous base. What do you notice here? What do you think they have in common? What do you guys think? What do they have in
The circle, like they have that the same.
Yeah, so so thymine and cytosine, they have one ring. See that? Right? And what do you notice about adenine and guanine?
They have 2 rings.
They have 2 rings, exactly. And so when we're looking at these molecules, right? Okay, we can identify these nucleotides based on their molecular structure. And so that's what you're going to have to do on the test. So you need to have diagrams of these for the test, okay, so that you guys can understand them, right? And so what do you notice every time you see a thymine nucleotide? What do you see on the opposite strand?
An adenine.
An adenine nucleotide, that's right. And every time you see a cytosine nucleotide, what do you see on the other side?
Guanine.
A guanine nucleotide, that's right. And so that's how DNA is constructed. Okay, they're just there. It's just a big molecule that's made up of these smaller molecules called nucleotides, right? And so then what ends up happening is, see these little things in the middle? Though, what do you think H bonds mean? What do you guys think? What do you think the H means stands for? You know what the H stands for actually, because
Hydrogen.
exactly. So what happens is, okay, thymine and adenine, what happens is their atoms are attracted to each other and they form these weak hydrogen bonds. So that's what allows DNA, right, to have two sides, because these two molecules kind of form these bonds. Okay, and it's almost, think of it like a zipper, right? Okay, it has the zipper has two sides and they come together in the middle. The two sides come together in the middle. Does that make sense, right? And so if you take a look at the molecule, you're always going to see an adenine on one side and the thymine on the other side. You're never going to see, like, for example, you're never going to see a guanine and you're never going to and you're never going to see a thymine on the other side. That's always how it's constructed. Okay? Makes sense a little bit? So as you're looking for the test, really the biggest thing is understanding that DNA, right, is a big molecule that's made up of these smaller molecules called nucleotides. And the only thing that changes in the nucleotides is what?
The nitrogenous bases.
Very good, the nitrogenous bases, exactly. So you gotta know. And so any questions so far on this? Any questions so far? Questions on this? All right. And so now here what it's showing you is, okay, thymine always pairs with adenine. So I always think of it like this is how I remember it. I always think of like apples, trees, apples on trees, right? Okay. Adenine always pairs up with thymine. Apples on trees, or you can if you're Hispanic, atole and oh no, I think it was like atole and tacos or something. I don't know. However you want to use it, right? Or ants and or no, I like apples and trees. That usually works, right? Okay, but just you got to have something in your head to remind you of those base pairing rules. That's what this is called. It's called base pairing rule. Okay. Okay, and so then over here, okay, if we go back, oops. Okay, if we go back, that's one base pairing rule. The other base pairing rule is, okay, so adenine always pairs up with thymine, and then the next base pairing rule is cytosine always pairs up with guanine. So the way I remember it, okay, is always, okay, you can always think of guacamole, right? And what do you always have with guacamole?
Chips.
Chips, exactly, right? So guacamole and chips, that's how you remember, right? And so on a test, okay, you guys should understand this idea that DNA is made up of nucleotides, and that and because it has two sides, on one side, if it has a cytosine, on the other side, it's going to have a nucleotide with a guanine on it. Does that make sense? All right, so let's give you one last problem. You ready? Here's the problem. So if you have DNA, right, and it has 10 nucleotides total, all right? So it has 5 on one side, 5 on the other. All right, write that down. Okay? All right. Um, you know that, okay, you did a little sample and you know that it has um 2 thymines and it has 3 guanines, like in total. Okay, got it? So the whole molecule has, okay, this and this. So how many adenines does it have? And how many cytosines does it have? So that's the question, right? So if you know that there's a DNA molecule, right? And you know that the DNA molecule has 2 nucleotides with thymine and 3 nucleotides with guanine. Okay, how many adenines and cytosines will it have?
2 adenines and 3
2
cytosines.
adenine.
2 adenines, very good. Because if there's 2 thymines, then there's got to be 2 adenines. And then how many guanines?
3.
3, exactly. Very good, right? And so the way this would work is we can write this down like this. So thymine, thymine, guanine, guanine, guanine. So I can write down the DNA strand just by using the letters. Because remember, what does the other parts change?
No.
No, right? The sugars and phosphates don't ever change. So we can just literally write DNA by just using the letters, right? And then on this side, okay. Now the order of this, it could be like the order of the letters, okay? It depends, and we'll talk about that. But just remember, if there was a molecule, right, that had okay, 5 nucleotides or 10 nucleotides total, okay? If 5 of them were these, then the other 5 on the other side would look like this. You see what I'm saying? Okay, let's try one more. All right, so let's say we have a DNA molecule, and this time it has, let's say, 6 nucleotides. So for example, guanine, cytosine, and I'm just picking random letters, okay? Thymine, adenine, and cytosine, okay? What would this side look like? What nitrogenous bases would be on this side? What do you think? So what would be here?
C.
Cytosine,
Cytosine, very good. Over here.
guanine,
Guanine, very good. Over here.
Thymine,
Thymine, over here.
adenine,
adenine,
Adenine, very good.
thymine,
Thymine, very good.
and
guanine,
And
guanine.
guanine, very good. See that? Okay. This right here, okay, would be the whole thing would be a piece of what?
DNA.
Very good, right? And how many nucleotides make up this DNA? How many nucleotides are in this DNA?
12,
Yeah, 12, exactly. Very good. 12 total because there's 6 on this side and 6 on this side. Does that make sense? Right? And so that's how this works, right? And so eventually in class, what you guys are going to do is you guys are going to use this model to do this, okay? Whoops. Here we go. All right. Whoops. So for example, here's see how I just built one DNA strand, right? And so in class, you guys are going to build your own DNA strand, right? Now the order of these letters, okay? It's like if how many of you guys are wondering, well, what's the order of the letters supposed to be? Like that's something I would ask and be all confused, right? How come I put a C here and an A there and a T there, right? Okay, how many of you guys are wondering that? Right? Okay, but asking me that question is like asking me this. What if I wrote this? I love chocolate. Why did I write the letters in this way, in this order? Why did I choose these letters and write and put them in this order?
Because that's how the words were created.
But what am I trying to do with these words? What am I trying to do with these words? It starts with a C.
Communicate.
Yes, I'm trying to communicate this to you guys, right? I'm trying to tell you I love what?
Chocolate.
So this right here is a code that I'm using to let you know what I'm thinking, right? Okay, and so that's the same, this right here is the same thing. The order of these nucleotides is gonna depend on what you wanna say, right? What do you want the code to be? What do you want the code to say? You understand? So so this right here, the order of these nucleotides is the code. All right, is the code. All right, and so we're gonna talk about that. But right now, do you guys so DNA has how many sides?
2.
2.
Good, right? So it has 2. So let me put the other side. Boom, it has 2. And each side is made out of what? Each side.
Nucleotide.
Exactly. It's made out of nucleotides. And what's the only thing that changes in the DNA molecule?
The nitrogenous
Nitrogenous
base.
bases.
That's exactly. Okay. Make sense a little bit or no? Did it help a little bit? Yeah? All right, all right. So that's good. That's good. So we'll talk about it. We'll talk some more next time. All right, so here we go. That's good. All right. So any questions? Do you guys have any questions?